Welcome back, everyone. So this week we're going to get into our Unit 2 lab on macromolecules. So this lab, again, if you haven't received your lab manual yet, we do provide the sheets for you. Okay, so we do have it linked right here. This is, I believe, the last week we do have these available for you. So do make sure that you have purchase the lab manual from the bookstore. If you haven't gone to the bookstore in person, then I suggest you do so this week so that you can just grab it off the shelves and pay for it in person, and then that way you don't have to wait for it to be shipped. In addition to the lab manual, this week we will also be using a supplement. So you'll notice that each week there will be a supplement as well. And this is just because the lab manual is designed for in-person labs, and since we don't have all the equipment at home, we have a supplement to go along with each of the labs. And so this includes like videos, video links and things like that that we can use to modify the lab so that you can complete it in an in-home environment. Okay, so in addition to using your lab manual or downloading the link here, you'll need to download the supplement, so either in Word format or PDF format. So just get in the habit of downloading the supplement as well. Okay, and you'll notice with the supplement, it goes along with the manual, so it'll go with the exercises. So you'll want to have them side by side. So for example, like if you open up the lab manual, it'll go through carbohydrates first with exercise 2.1 activity A. So you'll read through the part of the lab manual under activity A, but instead of actually doing the experiment, because you don't have the equipment, you'll watch the video instead. Okay, but you'll still answer the questions, and then again, you won't have the reagent and everything like that, so you'll, even though you're not doing the experiment, you'll see how it's set up, and then you'll watch a video showing what the results look like, so that you can actually write down what the results should be in your lab manual, so that you can get the right answers on the quiz and so forth. Okay, so just have that handy when you go through the lab this week. All right, so let's take a look at the lab. So this is what it looks like, lab topic number 2 on macromolecules. And it's broken down into 4 sections. So the first section will cover carbohydrates, second section covers the lipids, third section covers the proteins. So notice we won't be talking about the 4th macromolecule category, the nucleic acids. And that's mainly because it's in everything, all of our foods. So we can't really test for it because it's in everything. But we're going to take a look at how to test for basically simple sugars in our foods, starch in our foods, lipids, proteins in our foods. And then we're going to basically take some common household foods and test for the different macromolecules in those foods. Okay, so that's what the lab will be for this week. So let's start with exercise 2.1 from your lab manual. So you'll want to read through the information and the procedure, even though you're not going to be performing it at home. So just to give you an understanding of how the lab is set up. Okay, so these are some pictures taken from the lab manual, so you can see how you're going to be how the lab was set up with a certain reagent. This particular reagent is called Benedict's reagent, and so it was mixed with the sample and then it's heated up. And so you'll watch a video on showing you exactly how the experiment was set up for the students in the class. And then answer the questions, and then you'll watch another video showing what it looks like for a positive result and for a negative result. So that way when we test our food items later on, you'll know what a positive result looks like and what a negative result looks like for the presence of monosaccharides. Okay, so you'll watch that video. All right, and then make sure you take notes along the way and write the answers to the questions along the way. Okay. And then the second activity is for starch. So remember, starch is a polysaccharide, and so to figure out the presence of starch, we're going to use iodine. So iodine, basically, you're going to watch a video again on how the experiment was set up. Very similar to the Benedict's reagent, where they just drop the iodine into a test tube along with the sample. This time though, that you don't need to heat it up, and so it was, pretty quick experiment where it either changes colors or it doesn't. And then you'll watch that video showing a positive result versus a negative result. Okay, and then just make sure that you fill in the information, answer the questions in your lab manual. Okay. Moving on to exercise C. Exercise C goes into the hydrolysis of carbohydrates. So what does that term hydrolysis mean? Remember, hydro means water, lysis means breaking apart. So in this case, we're using heat as well as adding HCl, hydrochloric acid, to break sucrose apart. Okay, and oh wait, yeah, I think so. Yeah, sucrose, and then this one has starch. And so by doing so, we're going to see if we can break it down into the monosaccharides. Okay, and so starch, what is starch made of? It's made of glucose, right? So theoretically, if we break down starch, we should be able to test positive for the monosaccharides using the Benedict's reagent. So we should get a positive result if this experiment worked. Okay. Sucrose is a disaccharide, so if we break it down, we should also get that same result. So you'll see how the experiment is set up. You'll read through the information and the procedure, answering the questions along the way. And then there's a series of videos that show you how the experiment was set up, and then the results after several minutes. So you'll use this to help kind of fill in the chart in your lab manual. So what it looked like after 2 to 3 minutes, after 5 minutes, after 10 to 15 minutes, and then what the test results were at the end. Okay, and then once you've filled in your table, you can answer the remaining questions on page 39. Okay, so that kind of helps walk you through the whole experiment. And then moving on, in exercise 2.2, we're measuring for the presence of lipids. This one's a pretty easy experiment. So basically we're looking for if it's translucent or not when we put the sample on a piece of paper. So if you hold it up to the light and you can see through the paper, that indicates the presence of a lipid. So you'll see how that experiment is set up through a video. You can actually perform this experiment at home if you'd like, with just some vegetable oil and some water on a piece of paper. You can just watch a video showing what the results are. And then once you've completed the experiment or you watch the video, you can use the results to answer the questions on page 41 of your lab manual. And then moving on to exercise 2.3 on proteins. So proteins, we kind of go back to a more traditional experiment where we have our test tubes again. So this is something that you cannot test at home because you won't have the biurea reagent. And so this one, it's kind of the same as the iodine, where you put the sample in and then you put the biurea, and then after a few minutes it changes color. Okay, and so you're going to watch a video showing how it's set up, as well as what the results look like for a positive and a negative result. And then once you've watched the video and you've made your observations, you'll go ahead and answer the questions on page 42. And then finally, in exercise 2.4, we'll be looking at the presence of macromolecules in some common food. So the first activity is basically a separation of butter activity. So you'll read through the information about like clarifying butter, like how to clarify butter. And hey, you can actually do this at home if you want. Which is actually, you can take a stick of butter and actually clarify it if you want, but you don't have to. And so you can watch a video showing how the experiment is set up, where the butter is actually separated. And then you can watch videos showing how it was tested for the different presence of the macromolecules. So, how we tested it for, what it looks like when we tested just the whole butter, for the presence of monosaccharides, starch, lipids, and proteins. And then what happens when we tested the separated butter, the upper layer, for the presence of the different macromolecules, and for the lower layer. So that way we can kind of see, which part of the butter is, made of lipid, which part is made of protein, which part contains sugars, and so forth. Okay? So once you have watched the different videos and kind of filled out that chart on page 2.2, you can use the results to answer the remaining questions for activity A. And then finally we have the rest of the macromolecules, where we're going to test for different food items in activity B. So the experiment that we have videos for are 5 different food items. They're not these foods. I can't remember what foods she was able to test for. So you can just cross out the names of these food items and write in whatever names that Jennifer Bone was able to test for. And then you can watch, what happens when she, does the Benedict's reagent test, what happens to the iodine, the biuret, and the paper test results. And, you can put a plus sign for if it's positive for it, or a negative sign if it was negative. And then answer the questions on page 47. All right. So once you've completed the lab in its entirety and you've answered all the questions in the lab manual, using the videos and the supplement and everything, then you're going to download and complete the Unit 2 Reviewing Your Knowledge. Okay, so let me back out of this and let's go back to Moodle. Where did my Moodle page go? Hang on one second. Okay, so let's go back down to Module 2. And under our lab topic 2, we're going to you're going to be completing a Reviewing Your Knowledge worksheet that you're going to be turning in for points. You're also going to be submitting the following pages for basically check for credit, check that you actually did the lab. Okay, so these are the following pages that you're going to be submitting for credit. So page 36, 39, 44, 46 through 47 from your lab manual. Okay, so you can take pictures of those with your cell phone and then upload them through Adobe Scan that converts it to a PDF right away, or whatever other method that you have. Okay, so those will get submitted via this link, and then this link right here will have your Reviewing Your Knowledge worksheet. So this is where you're going to actually download the worksheet and use what you learned from the lab itself to answer the questions on the worksheet. And so these two are due by Sunday midnight, so ignore that right there. So it's Sunday midnight is when these two pieces are due. So the lab pages as well as the Reviewing Your Knowledge worksheet. Right? If you have any questions in the meantime, feel free to email me or stop by my office hours. Otherwise, good luck.